So there's some funky section numbering here, so I'm sorry. This is with the old book, chapter 43. Really, this is 10.2. We're looking at the digestion system. However, the digestive system, as interesting as it's going to be, is not a tremendous content part of the curriculum. You do not need to memorize all of the organs and what they do. Rather, you need to have a general idea of how cells interact really to maintain dynamic homeostasis. So we actually really want to focus on this part, the idea of fueling body activities. No matter what type of organism you are, you need nutrition for two reasons. You need nutrition to get energy, to be able to do work, and you also need your monomers to be able to build other necessary molecules. So while in an anatomy class you would be very, very focused in on the differentiation of the different organs and how they collaborate and cooperate, we are going to look at that. But you don't have to necessarily memorize all the names of the organs and exactly what they do. The main focus instead for us is the idea that we are consuming food for these two main functions. And so that's really what we're going to be focusing in on is how does digesting nutrition allow us to do these two things. So just real quickly, an overview of how food is digested. We have mechanical digestion, which is basically the grinding of food. And so this is going to be with teeth for mammals. When you look at other animals, they don't have teeth like we do. They might have different structures. Like insects have very different mouth structures, but they're not teeth. But many organisms have a way to grind mechanically food. Now when we look at digestion, we are mostly looking at multicellular organisms such as animals. However, we do have to keep in mind that whether you are a heterotroph and you eat food, or you are an autotroph and you make your own food, all organisms are going to be performing cellular respiration and they will need that energy fuel. So we are going to focus on the digestion of the mammalian system, even though there are lots of different ways to look at digestion. If you are a mammal, you use teeth to grind your food, but you use enzymes to actually break them into monomers. And one of the big keys that you need to be able to take away is the idea of compartmentalization, or compartments, environments, and dynamic homeostasis. Homeostasis. So this is a funny little cartoon that shows you that when you eat food, something is added to help break it down. And then more things are added to help break it down. And then some things are removed and some is waste. And so we do want to know that there are specific structures. This idea of cell differentiation, that you started as one cell, you underwent mitosis to make identical copies, but the cells differentiated. We now have different environments, compartments. So when you think about the digestive system, you do want to know that there are all of these different compartments and that they each have different jobs. Now when you first begin to eat, your teeth will do the mechanical grinding, but we do want to know that there are enzymes added here in the saliva. The esophagus is going to be the tube that leads down into your stomach. Now you'll notice I'm not writing anything here because you don't have to memorize what the esophagus does. but you should have a general idea that there is this tube that leads down into your stomach versus a tube that leads into your lungs. That's covered by something called the epiglottis, this flap, to make sure that food goes into your stomach, not down into your lungs. But once it gets into your stomach, we do want to know, just for general knowledge, that the stomach has two main actions. The first action is really storage. Most people think, and most biology or science teachers teach, that your food is digested in your stomach. And this is only true for proteins. Only proteins are digested in the stomach. Everything else is actually going to be digested in the small intestine. So what we do want to know is that the stomach is a place for storage. It is a compartment. And released into the stomach is going to be your acid. And the acid is mainly there to kill microbes. However, the enzymes that are there are going to have to be differentiated. Those enzymes that break down proteins, and only proteins, called proteases, they're going to be specific for proteins, and they're going to have to be differentiated to be able to withstand this acid. So you should be able to, at this point, be able to talk about a graph that looks at peak enzyme rates. And if I see an enzyme, a pH, that is really low, I might think that this is an enzyme that works in the stomach, versus an enzyme that does not work well in a low pH can't possibly be in the stomach. So what we're going to be seeing in this unit is a lot of applications to things that we've been learning earlier in the year. A key is this idea that you do digest foods in different places. Coming into the stomach is going to be the acid through the pyloric sphincter. Again, there's a lot of anatomy that we're not going to directly cover. This is maybe the most important part of our examining the digestive system, is that in your stomach, you are going to create something called chyme. The acid and the enzymes are going to mix everything into a very fluid source. And so entering the small intestine, chyme is going to be the dissolved nutrients and here we're going to see that carbs and lipids are going to be broken down. Again, you do not have to memorize where carbs are broken down and where lipids are broken down. As much as you need to know that at different parts in the mammalian digestion system, different enzymes are going to be active. So you don't have to memorize the word lipases. You don't have to memorize any of the names of the carbohydrate enzymes. You do have to know that they happen in different places. And so if I give you a chart of enzymes, you should be able to apply some prior knowledge. The key about the small intestine, we are going to have digestion. That is for sure. That's where the carbs and lipids are broken down. That is going to provide the function of digestion, breaking large macromolecules down into the monomers. But the bigger thing is really going to be absorption. If you're talking about a multicellular organism, like an animal, it's very important that we're able to break down these big nutrients into the monomers, but also get them transported throughout the whole body. So absorption is going to be where monomers are absorbed or transported. Let's use the word transported. Into the bloodstream. To circulate to the body cells. And so one one of the most important functions of the small intestine is we want to optimize or have the highest rate of absorption as possible. We want to absorb as many of these nutrients as quickly as possible. Well, in order to have a high rate of absorption, it's been found that having a high surface area will allow that to happen. So what we see with the small intestine is that it's actually small in diameter, but very long. So the small intestine is actually very long, but narrow. And we're going to see huge amounts of what are called convolutions or involutions, or this idea of really, you can just call them folds. And so the small intestine is going to be coiled up inside of your abdominal cavity. And because it's able to go back and forth, we have a lot of surface area in a small volume. But when we actually zoom in, and this is what your note card asks you to draw, when you zoom in on what those cells actually look like, we see that the cells themselves are packed on top of each other in a coiled way, and that even on the cells are the same structure. Now these are called villi. Villi are the folds of the arranged cells. And they tremendously increase the surface area to help with both digestion and absorption. So these folds help with digestion because there's more space for it to occur, and they help with absorption. So we have the villi, which are the arrangement of the cells, but then we have something called the microvilli, which if you look at this individual cell here, you can really see that on the surface of the cell membrane itself is what's called invaginated or involuted, meaning it has folds itself, and those are called the microvilli. And so this is actually a picture of the intestine, but if you zoomed in to look at the cells, you would see that the surface of the cells also has a highly increased surface area. And this is to maximize the rate of absorption. This is structure to function. These cells are differentiated. They are built a different way because of the expression of their DNA. And that allows them to perform a better job. The nutrients are actually being transported into the bloodstream. So when the blood arrives at the small intestine, it has a pretty low concentration of things like sugar. So arriving blood has a low concentration of nutrients. When the blood leaves, it actually has a very high concentration of nutrients. This leads us to the other organ that we really want to talk about when we talk about the digestive system, which is the pancreas. The pancreas is going to help create digestive enzymes. So some of the digestive enzymes will actually be manufactured here in the pancreas, but we're also going to see, as you should already know, that this is where we create and store, and that's really important. Not just create, but create and store insulin and glucagon, which together regulate blood sugar levels. Now I want to go back just for a second to talk about how does the sugar actually get into the blood. Remember, a monosaccharide is going to be slightly polar, and it's going to be fairly large. So we're actually going to have to use something called facilitated diffusion. We don't want to be using active transport because that takes energy, but we can use something called facilitated diffusion to help get the sugar into the bloodstream. Once the sugar is into the bloodstream, then we need to regulate the levels that are there. Now the rest of the digestive system we'll cover a little bit just as we wrap up in the next night, But the main part that you need to know is cell differentiation and this idea of cell communication and collaboration.